so we might start with the design of your study, Jean-Marc.
Yep.
Can you tell us about the research that you're doing and why it's so important?
Yeah, the research I'm doing, especially in this paper, is looking at the genetic variation of some genes and lines which has been discovered in the Tibetan Plateau, which is a part of China. Imagine how big the Tibetan area, which is probably 3 times of the whole of Britain and a third of Australia. So it's a very high latitude. Most of the areas above 5,000 meters above sea level. So those unique conditions caused the Tibetan barley to be very tolerant to extreme environment, such as salinity, drought, and cold. So this research is focused on salinity. As you may know, salinity is a very severe problem for Australia and across the world. It affects about 20% of the whole cultivated land, and nearly half of the land are more or less been affected by salinity. Salinity affects the plant growth and lead to reduction of productivity. So that's why it's so important.
So can you tell us about barley? Why is it that you've chosen to study it? Why is it so important?
Yeah, look, barley is a very important crop, both for feeding and for malting. And another reason for choosing barley is because barley is among the most salt-tolerant crops. It's much more tolerant than wheat, rice. So these are the reasons.
So we know that salt tolerance is important. So how can your research help us to develop more salt-tolerant crops?
Yeah, well, the genes discovered in barley were usually related to those we'll call homolog genes, which are similar in other crops like in wheat, in peas or other crops. So we're looking at the genetic variations and also try to identify genes from barley, which can always lead to the identification of those similar genes in other crops. And by doing so, we save a lot of time, and we can actually help to improve the salt tolerance of other more salt-sensitive crops. I think that's one of the main aims.
So now, Jean-Marc, we want to move on to the methodology. How have you gotten this study done? So would you be able to elaborate on how you've gone about conducting this study and collecting your data?
Well, yeah, it's actually a quite complicated process, and it takes us 1.5 years. This is mainly a PhD project. The whole experiment has 4 components. We look at the plant biomass, which is very important directly to the agronomical traits. And then we get into physiological traits. The 2 main components we're using is looking at the sodium and potassium content. As you know, salt stress is partly because of sodium, the high accumulation of sodium, which affecting plant growth. So we're looking at the sodium, and potassium is also very important. Then the third part is the molecular part. We amplify the CBF genes, which we had 3 of them, and in all the 188 Tibetan barley lines. We use those genetic information for further analysis. The fourth part is also we call in silico part, but it's very important. We're using a range of bioinformatic methods to look at the genetic associations and genetic variations of all those traits, and trying to identify the genes and looking at the differences of those genes in the salt tolerance of Tibetan barley. Okay, now I'm going to walk you through the paper and take a few graphs and tables as examples. For this research, we did the biomass analysis, which is the first section of the methodology. Out of the 188 lines, we discovered that about 70% of those lines are more tolerant than a commercial barley cultivar CM72, which is very exciting, especially from a breeder's point of view, because they see a great genetic resource for future improvement of salt tolerance in barley, especially for the current commercial cultivars. Most of them are not that tolerant to salt. So we found quite a few significant single nucleotide polymorphism. That means even it's the same gene in different lines of Tibetan barley, there are single nucleotide difference. That means some of those mutations, these are natural mutations, not transgenic, are actually helping the plants, the barley, to be more tolerant to salt. So we found about 13 of them. This will provide very important information for future breeding program. Let's move to figure 6. This figure shows the correlation between the relative dry weight and ionic content. As I mentioned earlier, sodium is very toxic to land plants. Most of land plants are not tolerant to sodium, so they cannot grow in seawater or half seawater or even a quarter of seawater. But those Tibetan barleys are actually able to grow to nearly half of the seawater salt concentration, about 300, 400 millimolar of sodium chloride. And we found that the sodium content in the leaf are negatively correlated with shoot dry weight. That means the shoot biomass are affected by sodium. And potassium is another important ion. Potassium is very important as for plant growth and regulating enzyme activities and for osmotic regulations. So sodium always competes with potassium for its role, and it's affecting normal plant growth. So we found the potassium concentration is positively related to shoot dry biomass. This gives us a very important information on how those ion content are affecting the plant growth, and this will provide some information for future barley breeding. So the identification of those CBF genes and their genetic variations in the 188 Tibetan barley lines gives us very important information. For example, they correlate with significantly lower sodium-potassium ratio and also the higher drying rate. That means in those lines and those genes will play a very important role for future improvement of salt tolerance in barley. In Australia, we have a very dry condition. Most of our agriculture are dry land agriculture, and our barley fields are not irrigated usually, and most of them rain fed. So in most of the conditions, the barley are under constant stress from drought, and in a lot of cases is the combination of drought and the salinity. So in the future, there is a possibility we can introduce those Tibet barley lines to the Australian farm breeders, then later pass down to the farmers, and it will be great to help to improve the current commercial varieties, which has a lot of good traits like a high feeding value, high malting value for making beers and whiskies, as long as we can use those genes to improve their salt tolerance of the current cultivars. So this study will be potentially very important for Australian agriculture. And in a broader case, all those genes identified in barley may be used for future breeding program in wheat and other major crops in Australia.
So thanks for that, John White. We just want to thank you for giving us your time. I know that you're a very busy man. You've got a lot of research going on. And as you were saying before, this is one part of a larger study. So you're busy right now with your PhD students and currently in your lab. What is it that you're working on right now?
Well, this is an additional question, but there are a lot of things happening in the lab. Currently, we're doing a project for the Department of Agriculture of Australian government to look at still salinity tolerance, but look at the different traits. We're looking at genes which controlling stomata regulation. Stomata gut cells are very tiny cells on top of leaf, and they regulate carbon dioxide and water in and out of the plants. It's very important. So we are close to the end to finish this project, and we'll probably have some very exciting results coming out.
Okay, well, we look forward to catching up with you when you finish that research. Thanks, Sean.
Yeah, thank you.